Good evening, everyone. Welcome to Tamali's classes. In today's class, I'm talking to you about operon. So, what is an operon? See, operon is a set of similar genes when placed together or when clustered together in an orderly manner. So, sometimes in the environment there are some changes, and bacteria, to adapt to these changes, they follow this control mechanism. Okay, so what they do, they cluster the similar genes together in an orderly manner. And one of the most common you have heard about lactose metabolism, or you can say lac operon. So lac operon is very, very common, and also about tryptophan operon. So today we'll talk about both the operons. Okay, so let's start. So first of all, we'll talk about who discovered the operon. So operon was discovered by Jacques Monod in 1953. And when we talk about lac operon is basically the lactose metabolism. Okay, so when lactose metabolism is occurring, you're using this some set of genes, then it's a lac operon. Okay, so what happens out here is that this lac operon, it encodes proteins, or you can say there are many genes out here which will encode protein, okay, allowing the bacteria to use lactose as an energy source, especially when lactose is present. Okay, and this system is a negatively controlled inducible system, and it helps in regulation of the expression of three structural genes. So what are the three genes? Gene Z, gene Y, and gene A. So here you can see the diagram of lac operon. It's very simple. So see here. So this one in absence of inducer. Okay. So first of all, we'll talk about the players of lac operon. That means we can say the participants of lac operon. So first of all, we have out here regulatory gene. Okay. So you can see out here this is regulatory gene. Fine. So here you can say it's I, also known as gene I. Okay. So or you can say lac I. So here again we have promoter. Promoter is the beginning. Okay. It's the beginning of the operon. So it starts the operon. Then we come to the operator part. Operator is just placed near to the promoter. You can see this is the operator out here. This is the operator. So you can diagram is like this. Okay. It's like this: regulator, regulatory. Then we have out here promoter. Okay. Then we have operator. And then we have three structural genes: Z, Y, and A. Okay. So the order is like this. So what happens? Regulatory gene, it produces either it produces a repressor protein. Okay. So regulatory gene, it produces a repressor protein, and promoter out here, it is the place, you can say it's the starting point of the operon. Okay. And operator is that site wherein either the activator or the repressor will bind. So the state of the operator determines whether transcription will occur or not, whether translation will occur or not, whether proteins will be formed or not. Okay. And whether the genes, or you can say the gene expression, will occur or not. Fine. So I told you about regulatory gene out here. So it will produce the repressor protein. Then promoter I have told you. So promoter is the place where RNA polymerase is present, where it is recognizing the where it will recognize and it will bind to the operator and begin the transcription. Okay. Then we have the structural gene ZYA. So out here, this Z gene, it codes the beta galactosidase enzyme. Okay. So Y out here, it codes for the galactoside permease enzyme, and for the A, this codes for the thiogalactoside transacetylase enzyme. Okay. So what happens out here is that when lactose is present, fine. So this lac Z enzyme or lac Z gene, what does it do? So it produces this beta galactosidase enzyme. So when lactose is present, this enzyme helps in the breakdown of lactose into glucose and galactose. Okay. Then what does Y do? This permease enzyme, it will help in the transfer of the lactose using the proton gradient. And this lac A gene, it will help in the transfer of acetyl groups, okay, to the thiogalactoside. Now, this is the function of structural genes. Mostly this one out here, lac A gene. This function is not known. Okay, till now it is not known, but it is said that it helps in the transfer of acetyl groups. Okay. Now let's come to the process. What happens out here? Now suppose lactose out here is not present. Okay, suppose lactose is not present, or you can say there is no inducer out here. What will happen? This regulatory gene will produce the repressor out here, and since there is no inducer, since there is no lactose, what will happen? This repressor will go and bind to the operator, and it will block the operator. So once the operator is blocked, what will happen? this RNA polymerase, it cannot move or it cannot slide through the operon. Okay, it cannot slide or move through the operon. Therefore, no transcription will occur. Okay, out here, therefore, what will happen? There will be no transcription of the operon. The genes will not be expressed. Fine. Now, coming to the part when the inducer is present. Now, suppose inducer is present, or you can say lactose is present. Okay, what will happen? Out here, a regulatory gene will produce the repressor protein, but what will happen? This lactose will bind to the repressor. It will form an inducer. No, example is allolactose. Okay, allolactose is produced out here, which is an inducer. So allolactose, what will it do? It will inactivate the repressor. So remember this thing, that allolactose is an inducer. Okay, it will go and bind to the repressor protein that is formed by the regulatory gene out here. See, this is a repressor protein. So it will bind to the repressor protein and it will block the, or you can say it will inactivate the repressor protein. When the allolactose is inactivating the repressor protein, therefore what will happen? The repressor protein cannot bind to the operator, and operator is free. So when it cannot bind to the operator, what will happen? RNA polymerase will come and bind to the operator. It will recognize the it will recognize the sequence. Okay, it will go and bind to the operator and it will start the transcription, or you can say mRNA transcription out here. Thus, and again it will lead to the formation of the protein. Translation will occur from mRNA translation will occur, and it will form the protein. Okay, so this is occurring out here. Fine. I hope you have understood this one, this process out here. Fine. Now, moving on to the next part. Now see, so I hope you have understood this thing. So when inducer is present, only then lactose metabolism will occur and lac operon will be active. But when there is no inducer, then what will happen? Repressor protein will bind to the operator and block the expression of gene. Okay, it will block the operator and thus there will be no expression of gene or regulation of gene expression. This is all about your LAC operon. Now coming to this tryptophan operon. What is happening? This is just the opposite of LAC operon. What will happen this one out here? So this is tryptophan operon, wherein it will encode some enzymes which are required for biosynthesis of amino acid tryptophan. Okay, from the aromatic precursor known as chorismate. So out here, similarly, just like the LAC operon, there are some group of genes which are clustered together and which will help in the production of tryptophan. Okay, for the biosynthesis of tryptophan from chorismate. Now again here there are different structural genes. That is, it will be like TRP A, B, C, D, E. So what does this what do they signify out here? You can see. So tryptophan A out here, it signifies tryptophan synthase. Okay, with the alpha subunit. Tryptophan synthase, beta subunit for B. For C again we have out here N5 phosphoribosyl anthranilate isomerase or indole 3-glycerol phosphate synthase. For tryptophan D we have here anthranilate synthesis with component 2, and tryptophan E is out here anthranilate synthetase with component 1. So these are all the 5 structural genes of tryptophan operon. Now if you see out here the structure, this is again very easy, just follow the process out here. Now suppose tryptophan is present. See, this process is for the biosynthesis of tryptophan. So if tryptophan is already present, then is it required? No, it is not required. Therefore, gene will be off out here. Okay. So if tryptophan is present, then the regulatory gene will produce repressor protein, and then the repressor protein will go and bind to the operator and it will block it. Okay. It will block it. Therefore, no regulation of this outer, no biosynthesis of tryptophan is occurring. Okay, so transcription is completely blocked. Okay, now suppose there is absence of tryptophan. Now you need tryptophan because in the cell there is no tryptophan. Then what will happen? The gene turns on. Okay, the gene turns on. Out here also repressor will be produced, but since there is absence of tryptophan, what will happen? This repressor will not bind to the operator. Okay, the repressor will be dissociated from the operator, and what will happen? The transcription will occur. Okay, so this, all the enzymes for synthesis of tryptophan will be active and they will help in the production of tryptophan. So this tryptophan operon is also known as a repressive operon. Okay, remember this. Why? Because out here tryptophan acts as a repressor. Because when it is present, it will not help in the transcription. But when it is absent, only then it will let the transcription to occur. Okay, I can say yes, let the transcription to occur and formation of tryptophan. Fine. Therefore, tryptophan operon is also a repressive operon. This operon was also given by Jacques Monod. Okay, so this is how the tryptophan operon works out here. So see out here, suppose there is no tryptophan, what will happen? So this promoter out here, this RNA polymerase will transcribe the structural genes. They are all transcribed. mRNA is produced, and mRNA will again help in the translation process and help in the protein formation. Okay, so this will help in synthesizing the tryptophan. Again, when tryptophan will increase, again gene will become off, and again same thing, transcription will be blocked. Okay, so this continues. This is the working of tryptophan operon. Fine. Therefore, what do you understand by this? That when the tryptophan is high, then no transcription. And when it is low, transcription will occur. Therefore, this tryptophan operon is regulated by the amount of tryptophan present. And what did we see in LAC operon? That the state of the operator will determine whether the transcription or whether the regulation of gene expression will be there or not for LAC operon. Okay, because if the activator or repressor binds to the operator, only then we can decide. If the repressor binds, no transcription. If the activator binds, then there is transcription. Okay, so this is all about your operon. I hope you have liked it. Do click on the like icon if you have liked it, and share it with your friends, and do subscribe my channel for more such videos. So see you next time. Bye bye.